My extreme vertebrate was the yellow-rumped leaf-eared mouse. Taxonomically, the Phyllodes Zinthropicus belongs to the Mammalia class. It is then following down to the Rodentia order into the Chrysetidae family. This family includes other members like true hamsters, voles, lemmings, and New World rats and mice. The yellow-rumped leaf-eared mouse gets its namesake from its rounded leaf-shaped ears and from its yellowish-brown guard hairs that contribute to their overall gray-brown or sand-colored coat. Like many rodents, camouflage is an important survival mechanism for them because it helps them hide from their primary predators, which are raptors that use their eyes to spot prey. As a result, their thick coat can vary in color depending on the season to best camouflage within its habitat. The mouse is found in the Andes mountain range of Argentina and Chile. These mice are found in saxicolous environments, meaning that they live among rocks. In 2020, this rodent was found at an elevation of 6,739 meters, which is over 22,000 feet at the summit of Yuyuyaco volcano, making it the world's highest dwelling mammal. Unfortunately, not much food exists at this elevation, making the mice opportunistic feeders, eating any vegetation, insects, seeds, and fruits that are available. Behaviorally, these mice are able to go into torpor when body temperatures drop down low enough to help conserve energy since non-shivering thermogenesis is expensive. To avoid predation, we also observe different foraging habits. In 2001, an experiment was done by Kramer and colleagues where they observed an increasing foraging activity in darker environments compared to those in brighter environments. This helps to suggest the idea that they forage at night to avoid being seen by predators overhead who might be looking for food. As we climb higher in elevation, the air temperature decreases. These mice were studied by Nespolo and colleagues in 1999, where they observed metabolic plasticity within differing populations at different elevations. Populations that were adapted to higher elevations had higher maximum metabolic rates and rates of non-shivering thermogenesis. Upon acclimation of low elevation mice to higher elevations, they observed a 94% increase in maximum metabolic rate with a 49% increase in non-shivering thermogenesis. This is because when exposed to the cold, the sympathetic nervous system of the mouse releases norepinephrine uh, in brown adipose tissue. This hormone signals a pathway that allows protons to be pumped into the matrix of the mitochondria, creating heat as a byproduct. However, this project is metabolically expensive. This means that they are able to live at colder environments because they have higher rates of non-shivering thermogenesis and higher maximum metabolic rates. And this allows them to acclimate to their seasonal climates easily, but it comes at a high energy cost. Phyllodes Zinthropicus was also found to have increased carbohydrate use for aerobic exercise compared to other Phyllodes species. Research has found that they have adapted a metabolic mechanism that uses glycogen stores first upon stoichiometry analysis of the mice. They also found that they were able to get 30% more ATP per mole of oxygen in lipids or in carbohydrates than lipids in this Phyllodes Zinthropicus species.